Hello, everybody. Thank you for joining me today. I hope you're having a pleasant event so far. My name is Beate Kern. I'm a product manager at Nanotemper, and it's my pleasure today to talk to you about how you maybe can improve your biopharmaceutical development by applying Prometheus to get you stability measurements spanning the entire product lifecycle. I don't have to tell you that biologics are important. I also don't have to tell you that biologics are becoming more and more important all the time. But what I want to illustrate by using this slide is that they have been around for a while now, and we've had some time to learn from the past. One thing that we have learned is that in early stages of biologics as a whole, a lot of focus was put on activity, so active molecules that performed the binding that they were designed to do, for example. And not a lot of attention was paid to their stability, because stability was considered a parameter that can be engineered in or formulated in later on. Well, it turned out that wasn't always true. So a lot of molecules were advanced into later stages that didn't have the stability necessary to be successful. These days, researchers are finding a balance between activity and stability and making sure that they look at stability even early on to only advance those molecules that are actually worth investing in. This means that protein stability is now getting considered pretty much anywhere in the development process. For example, very early on when looking at developability, then characterizing the molecule at any step, looking at pre-formulation or formulation, later on in process development or in production, and then even after that's been established in comparability. Protein stability is a pretty complex topic though, so it's not just one thing that you measure typically, it's a pretty complex matrix. So there's different aspects to it. I only want to highlight a few. For example, there is the properties of self-interaction and cross-interaction, so how likely a molecule is to interact with the same species or a different one. There is hydrophobicity, there is colloidal stability, meaning the propensity to aggregate. There is also thermal stability, and of course chemical stability. So all of these are, as I said, different aspects of the same thing, and they're not all of the aspects there are, but already here you can see that this is a topic that needs to be looked at from different perspectives. So what you want is instrumentation or technologies that give you clear answers on these points, because if you get vague answers for several factors, that's not going to help you at all. You won't be able to base any decisions on that. But if I say clear answers, what do I mean? Let's focus on two aspects for today, colloidal stability and thermal stability, because those are the aspects that Prometheus can help you with. And as I said, clear answers, what does that even mean? What does an instrument have to be able to do for the answers to be clear and actionable? Of course, the measurement needs to be specific. So you need to be able to distinguish the signal of the biologic from the buffer or matrix. The limit of detection needs to match your applications. So no matter whether you're working with very high concentration biologics early on in development or very potent molecules, or whether you're working with very high concentration biologics, it doesn't matter. The instrument needs to be able to handle them. It also needs to be sensitive so that you can identify domains with subtle signals, not just the big stuff. You also need to be able to see the small stuff. You want, of course, a high level of accuracy and precision, meaning that you report the true values, but you also can report them in a repeatable and consistent way. There can't be variation from day to day or instrument to instrument or site to site or whatever. And finally, you need excellent resolution so that you can actually distinguish domains or proteins with similar properties. So I want to walk you through 3 case studies in this presentation that highlight how the Prometheus addresses all of these 6 points and excels in all of these 6 things. And the 3 case studies that we're going to look at are located in very different stages of development. 2 are located pretty early on and 1 is later. I want to actually walk through them in reverse order, so we're going to start with the comparability study. This is a study that came out of Amgen very recently, and you can really see that they looked at specificity, sensitivity, accuracy, precision, limit of detection, and resolution. So all of these 6 important points, and we'll see how that made a difference there. Now, of course, a comparability study is something you conduct if you change your production process, either because you change the process itself or because you transfer to a different site or something like that. Comparability is important because you need to make sure that the change in process hasn't affected your drug product in any way, or its safety or efficacy or all of these properties. So this is a really regulated process that needs to be validated very, very thoroughly. And this is exactly what this group of researchers did. So they went ahead and validated Prometheus itself, whether it's even suited to this kind of application. They looked at specificity of the signal. Here, as an example, we are seeing the unfolding signal of a BiTE molecule, a bispecific T-cell engager. And you can see very clearly that it's easily distinguishable from the signal of the formulation or buffer, which there isn't any. So that's very, very clear. The group also looked at accuracy by comparing the Prometheus results to what they had previously traditionally used for this application, which is differential scanning calorimetry. And again, you can see a high level of agreement between the two, so accuracy is proven as well. On this slide, I want to show you how they actually looked at several things at once. So we're looking at precision and at limit of detection in the same data set. They tested a lot of different molecules. Here's all the antibodies that they tested. And by highlighting just 2 of the samples, you can see how it also looks at the concentration range. So we are looking at one antibody at a concentration of 50 microgram per milliliter and the other at 74 milligram per milliliter. So that's 3 orders of magnitude difference between the two. But still, repeatability and precision is not compromised, even though you're working at pretty extreme concentrations. The Prometheus can actually go even lower and even higher too. So by repeatability, we mean the variation that you can expect between measurements like this. They measured all of these samples in 7 replicates. So among those 7 replicates, for all of those antibodies they tested, they got a repeatability below 0.2 degrees Celsius. So that's how precise you can expect your results to be on Prometheus. And then in a similar vein, they looked at not just the whole molecule, but at the different domains within the molecule, for example, Fab domain, CH2 and CH3 in an antibody, or at other types of molecules as well, like the BiTE molecule. And they found a repeatability of 0.3 degrees Celsius or below for all of them. And now this, of course, tells you about the excellent resolution you can expect on the Prometheus, because that's what you need in order to be able to distinguish these domains and to make sure you're looking at one or the other and you're seeing both and you're seeing everything that's there. Moving on to the second case study. This is a study that was conducted at Janssen, also published very recently, where they use Prometheus to characterize a biologic and engineer a better variant of it. We are looking now at a transgenic animal-derived antibody, which had some rare somatic hypermutations that compromised the stability of this molecule. So they engineered those mutations in order to improve the stability, and then validated those measurements with thermal and chemical denaturation measurements, and also related that to storage and accelerated degradation data. So the molecule we're talking about was exhibiting 4 of these mutations. You can see them shown here on the left. The molecule highlighted in orange is the, let's call it original, so the molecule that they originally discovered, and that had all of those 4 mutations, and then they looked at the amino acids that are usually found in those positions and mutated them all back to those. So they engineered 10 variants that are all listed here, where individual positions or combinations of positions are mutated. And the one that proved best was actually one that is now highlighted in green, which has all of these points mutated back. You can see that it performs better with regards to expected immunogenicity, but also, of course, thermal stability. It has a much higher Tm, and aggregation, so the temperature at which it starts aggregating is also much higher than from the starting molecule. They then, as I said, went ahead and validated that. They did chemical denaturation experiments, also on the Prometheus, by the way, where they were able to show that the engineered variant in green showed a higher stability compared to the original one in orange. And then further along, they stored both of these molecules for up to 28 days at 4 degrees and 40 degrees to see how much aggregation would happen over time. And again, you can see the green engineered molecule behaving much better, showing a lower percentage of aggregation than the orange original molecule at both temperatures. So that's a great example of how you can use thermal stability and chemical stability data to validate these things that you originally came up with, of course, through computational studies. I want to highlight that this group used an automated version of Prometheus, as you can see on the right here, but I also want to mention that this is a modular automation, so you can start with the manual instrument and scale up as you need. For the final case study, I want to show you an example of developability assessment. So this is a study from Merck, where they looked at a large set of potential candidates, did a comprehensive and thorough stability characterization using several different methods, and then took all of that data, looked at correlations between those individual measurements, Prometheus and others, and did a combinatorial analysis to look at this complex stability matrix that we talked about in the beginning to identify the best candidates early and to make sure they have no liabilities that prevent moving them forward. So here's an overview of the workflow that they were using. As I said, high throughput discovery and data generation on the left. Then they set up a system of really managing that data, because that's a lot of data, so that you can look at individual samples and the correlations and really identify all of those connections and connect the dots, basically. And all of that data was used to influence what they did, what they moved on into development and what they did in development, and how they really designed those molecules. Here's just a map that I just want to show you to highlight how many different types of measurements were run, how many different parameters were compared. This shows you the correlations between them. So the red spots highlight positive correlations, the blue ones highlight negative correlations. But if we take only those that are really interesting, so the strongest positive correlations, and pull them out into a table, this is what we get. And I've highlighted in yellow those that show a correlation between a Prometheus measurement and another one, so a different technology. Of course, there are also correlations between the individual Prometheus parameters. For example, thermal stability and aggregation may correlate, or do often correlate. But there's also correlations found between the melting behavior and isoelectric focusing detected isoelectric point of the protein, and also between the Prometheus data and UP-sec data, so monomer content data, which of course was not necessarily expected, so it's not surprising to see it, but it's great to prove it, because that means you can use one as a predictor of the other. What they then did was they clustered all of that data and found 3 clusters of proteins. The cluster number 3, the lowest one shown in the table here, being the one that showed the most optimal characteristics, because it had the highest monomer content, the highest melting point, as shown by Prometheus, the lowest tendency to self-aggregate, as well as the lowest content of high molecular weight species, and the highest isoelectric point. So this is really a good example of how you can look at protein stability from all of these different perspectives using Prometheus, but also other methods, and connect that data. And again, that data is not going to be useful for you if it isn't high quality data, but Prometheus delivers. So I think looking back at the 3 case studies, we saw very convincingly how Prometheus is specific, sensitive, has excellent resolution, accuracy, and precision, and gives you limits of detection that are really useful for you in whatever application you are. What I haven't necessarily mentioned yet is that Prometheus enables you to run those measurements in less time and with less sample than other technologies. You can look at more types of molecules and at a broader concentration range. We have talked about that, and also that it's automatable. And to kind of explain a little bit more about those last few points, I want to explain how the Prometheus actually works, because that'll really explain a lot. So what the Prometheus actually measures is the intrinsic fluorescence of proteins, their tryptophan and tyrosine residues specifically. This means we don't need a label. This is a label-free technology, no dye involved. These residues become exposed to the buffer when the protein unfolds, and that's what we can see because their fluorescence properties change upon unfolding. So we basically watch the molecules unfold while we heat them or while we add detergent. At the same time as this thermal unfolding experiment, we can detect aggregation happening in the sample with the back reflection technology. And the way we do it is actually pretty simple. We shine light into the sample and we see how much of it gets reflected back. And the more aggregation there is, the more light gets scattered and doesn't come back to the detector. So that's a really simple setup, but it gives you powerful information because it tells you at which temperature your molecule starts to aggregate. And this, as I said, this colloidal stability data right here and the thermal unfolding data from the previous slide, these are measured in the exact same experiment on the same sample. So there's nothing extra you have to do. And I also want to show you how this is actually set up. So the Prometheus uses glass capillaries to hold the sample. Each capillary holds 10 microliters, so really not a lot of sample that you need. And those capillaries are on a plastic scaffold, so what we call a capillary chip, because that enables really easy handling of the capillaries. You can load directly from plates, and this type of thing, the chip can be handled by a robot, so that enables automation. There is a thermal element in the instrument that heats the samples at a ramp that you specify and in a temperature range that you specify, anywhere between 15 to 95 degrees, and with a slope of anywhere between 0.1 degrees Celsius per minute up to 7 degrees Celsius per minute. And even at the high heating rate, you still collect many, many data points, which means you still have high quality data that is a solid foundation for your research. The way how we collect that many data points is that the fluorescence detector continually scans the samples and it takes a reading every 3 seconds. So every 3 seconds you get a data point. And that really is what translates into that high quality data. So I hope I was able to show you how the Prometheus can help you improve any biopharmaceutical development workflow, because it's able to provide you with the stability measurements that you need at any point of the product lifecycle with high quality data and reliable results.